Thank you to the organizers for inviting me. My name is Vikas Bansal, and I'll be talking about methods for detecting genetic variation in genes with high sequence homology in the human genome using short read DNA sequencing. Okay. So I think last few days we already have heard a lot about how DNA sequencing is now really cheap and ubiquitous. And this has happened gradually over the last about 15 years, starting in around 2007 when these sequencing technologies started to emerge. And today Illumina is the dominant technology for sequencing human genomes. And this technology basically generates short reads about 100 to 300 nucleotides long, and you can sequence a human genome to about 30x coverage for about $600 as of today. And since sequencing is so cheap, a lot of studies of human genomes have been conducted trying to find disease-causing variants or understand human population variation. And as a result, we have large databases of human genetic variation. A prime example of which is GNOMAD, the Genome Aggregation Database. And this database, as the most recent version, has variant data from more than 700,000 human exomes and genomes. And at the same time, we've also seen a rapid kind of steady increase in the number of phenotypes for which we were able to find the genetic basis. One common feature of all these human genome sequencing is that the standard paradigm for analysis is to align the reads to a reference genome, because that's the best you can do with short reads. And this is problematic because the human genome is repetitive. In particular, we have this class of repeats known as segmental duplications or low copy repeats that cover about 7% of the human genome. And these are problematic for short read sequencing because if you look at the illustration shown on the right here, if you have a sequence, a long enough sequence G that has two very similar copies in the reference genome, then if you do short read sequencing, after sequencing, when you try to align the reads back to a reference genome, you cannot really decide whether to align the read to G or its duplication. So therefore, it's very difficult to do any kind of variant calling in these regions. In 2016, a group at Harvard did this kind of sequence similarity analysis of the human reference genome. And what they found was that almost 2,000 genes, that's about 10% of the genes in the human genome, have at least one long enough stretch of sequence homology that is problematic for these short read sequencing. And among these 2,000 genes, about 50% have more than 75% of their exons that have high sequence homology to at least one other region in the genome. And this kind of is not really appreciated when people do exome sequencing or whole genome sequencing, because most of these regions tend to the reads mapping to these regions have low mapping quality, and they automatically get filtered out during the downstream analysis. However, we are interested in trying to tackle specifically variant calling and genetic analysis in these genes. And the prime motivation is that these genes are not junk, although they are duplicated, but they're still medically and biologically relevant. And here's a list of genes compiled again from this 2016 study that shows you a list of genes that are associated with rare and some cases common human diseases. So roughly about 193 of those 2,000 genes have some association with human disease, and about 85 of them have association with like Mendelian disorders. And you can see there are some examples like PMS2 here is associated with a germline syndrome of cancer called Lynch syndrome. And there are many other genes associated with both childhood diseases as well as adult onset diseases. And a very well-studied example of a duplicated gene or a gene with high sequence homology in the human genome is this SMN1 or survival motor neuron gene that's located on chromosome 5. The entire region, including this gene, is duplicated, adjacent to it, and the sequence similarity between SMN1 and its copy SMN2 is more than 99.8%. So what that means is that SMN1 and 2, they only differ by 8 nucleotides across the entire gene and only by 1 nucleotide in the coding region. So for all practical purposes, they're almost identical. And SMN1 and 2, this relevant because in 1995, well before even the human genome was sequenced, SMN1 was shown to be the causal gene for this rare recessive disease called spinal muscular atrophy, which basically causes muscle and neuron wasting. And interestingly, both SMN1 and 2 are functional, but it's only primarily SMN1/2 generates more functional protein than SMN2, and therefore the copy number of both SMN1 and 2 is relevant for predicting whether someone will have spinal muscular atrophy or whether it will be severe or a mild form of the disease. And if we look at the NOMAD database that I talked about earlier, and we look at this SMN1 locus, we see that basically it's completely absent in terms of variants in this large database. And the reason being that because the sequence of the two genes are almost identical, all the reads essentially have zero mapping quality, and you cannot call variants with the standard variant calling tools. So because these genes are so well studied and clinically important, some group researchers have tried to address the challenge of being able to estimate copy number. So in the case of SMN1, a group at Illumina developed this method called SMN1 copy number caller, which is basically designed to estimate the copy number of SMN1 and SMN2 using whole genome Illumina sequence data. And the key idea behind this method is that since the two genes are identical, we can basically collapse the reads mapped to the two genes together and just consider the read counts for the two genes together and estimate the aggregate copy number for SMN1 plus SMN2. And once we have the total copy number of the two genes together, then we can use the small number of positions that differ between the two genes to estimate the copy number for SMN1 and SMN2 specifically. And they were able to show that this method works well for estimating copy number of SMN1 and SMN2, and so it's a useful strategy for handling these genes. Another similar approach was developed for this gene called PMS2, and PMS2, as I mentioned earlier, causes Lynch syndrome. And in this case, the mutations that cause the Lynch syndrome are not copy number changes, but SNPs, and short indels. And PMS2 is also duplicated, but only the 3' end of the coding sequence is duplicated. And so in this paper, the idea was that you could basically adopt the same approach as the SMN1/2 in this copy number caller paper, and basically collapse the reads mapped to the duplicated region and do variant calling using the aggregated reads. And then using long range PCR, one could identify whether the variants identified are located in the PMS or the pseudogene copy of PMS2. So these kind of these two studies kind of showed that it is possible to look at genetic variation in these genes, but obviously you have to do something beyond what standard pipelines do. And although these methods they work for these specific genes, but if you think about the general problem, like can we build a general variant calling and copy number caller for all such genes that have high sequence homology, there's a number of challenges that come up because of the nature of these genes. First, obviously the biggest challenge is that a lot of the reads don't map uniquely, so that's kind of already obvious. Another kind of challenge that's unique to these genes is that there is frequent sequence exchange between the different copies of these duplicated sequences because of either gene conversion or recombination. And this has been observed for many genes. So here I'm showing you an example for the PMS2 gene. Because the two copies are located very close to each other, there can be non-allelic crossover generating these hybrid alleles or resulting in gene conversion. And so the consequence is that we cannot trust the reference genome to distinguish between the gene and its homologous copies. And I'll explain that further when we talk about the methods. Another complication is that many genes have extensive copy number variation while others are pretty much fixed in copy number. So that's very important for variant calling, so you cannot just assume that these genes are diploid or like have a copy number equal to the reference. And finally, some genes have a very complex duplication structure where some part of the gene is homologous to one sequence while other part of the gene is homologous to a sequence located elsewhere in the genome. So you have to kind of account for that if you want to analyze these genes. And around 2017 we decided to kind of work on this problem to basically build a framework for being able to do variant calling and copy number calling in these genes. And here's basically kind of the list of the problems that we're trying to solve. First, as I said, since these genes are vary a lot in copy number, our first goal was to estimate copy number of any given gene that has high sequence homology using whole genome sequence data. And we've developed a tool called parascopy for that, and I'll talk about that in detail. And we've also developed a variant caller, parascopy VC, which basically adopts the same approach that I mentioned for that PMS2 gene, but it works for pretty much any gene in the human genome that has high sequence homology. And another challenge related to these genes is that for many genes like SMN1, we can only do aggregate variant calling, which means that we cannot distinguish whether the variant is present or located in SMN1 or SMN2. So therefore, the question is this information useful? Can we do disease association analysis with these aggregate information? And I'll talk a little bit about that also. And finally, we've also worked on extending these methods to work with exome data, which presents another challenge because of the significant variation in read depth across the different exons. So I'll go into detail about the copy number profiling method, and before I do that, I just want to provide some basic definitions for our method, so that are relevant to these genes. So consider a gene that has two copies in the reference genome, which we label as copy A and B. Now any given sample, because we're talking about human genomes, if there is no copy number variation, we expect to see 2 copies of A and 2 copies of B. And in this case, we say that the paralog specific copy number or paralog CN is basically 2, 2. So 2 copies of copy A and 2 copies of copy B. And then the aggregate copy number, which is the sum of the different copies, is 4. And similarly, if you have individuals with 1 copy of copy B, then the paralog copy number is 2, 1, and so on. And the reason this is important is because the way we estimate copy number is first we estimate aggregate copy number, and then we use that to estimate paralog specific copy number. And for some genes, we cannot estimate paralog copy number, so it's aggregate copy number is all we can get. Another important concept is what's known as paralog sequence variants or paralog specific variants, or PSVs for short. So if you look at a gene with 2 copies, then if the sequence of the 2 copies is not identical, there are some differences between the 2 copies, then each such position is referred to as a PSV. So in this example shown here, you have this one position where copy A has the G nucleotide, whereas copy B has T. So these are important because these are the only positions that can be used to distinguish between the two copies or to determine whether a sequence read was sampled from copy A or copy B. Now we have this, we introduced this concept of a reliable PSV. So reliable PSVs are those that are kind of fixed in the population, that don't show any very little variation in their genotypes across a population. So if we look at genotypes for this PSV across a large sample of individuals, we would see that most individuals have both copies of copy A have a G, and both copies of copy B have the T allele at that position. So what this means is that these PSVs can be used to distinguish whether a read originated from copy A or copy B. And in contrast, we have the opposite case of an unreliable PSV. These are PSVs where they coincide with a polymorphism, and so therefore you see the even copy B has the G allele present, and copy A also may have the T allele present. And so these PSVs are not very useful for deciding whether a read originated from copy A or copy B. So this method parascopy for estimating copy number is basically a two-step method. So first we estimate aggregate copy number, and then given the aggregate copy number, we estimate paralog specific copy number by identifying reliable PSVs using a population, using multiple samples from a population. So I'll go over the different steps of the method, and its first step is to basically aggregate the read depth across the different copies. And all the examples I'm showing you are based on 2 copies, but the method works for any number of copies. So even if you have 7 copies of the gene in the genome, the method works. So what you do is basically you map all the reads from all the copies to one single copy. So in this case, we are mapping the reads from copy B to copy A. And so the advantage of doing that is that basically mapping errors become redundant, because we don't need to worry about whether a read came from copy A or copy B. And the next step is something that's very similar to standard methods for copy number calling, which is that we convert the aggregated reads to read counts in like windows across the region of interest. So the method is given a locus of interest, and across the locus it will divide it up into windows of a fixed size, and for each sample and for each window it will calculate the aggregated read depth across for each sample. And here I'm showing in this plot on the left, we're showing the normalized aggregated read depth, where each dot is basically one window, and the normalization is done using read depth from unique regions of the genome. And we have here 6 samples where we have the aggregated read depth, and our goal is to convert this aggregated read depth into these copy number profiles. And for that we use a standard HMM, where the read depth, basically the emission probabilities relate to the read depth, and then the transition probabilities correspond to the change in copy number events. And the only kind of unique thing here is that the transition probabilities are estimated jointly for all samples, and therefore we run the HMM for each sample multiple times. And so as a result, if some sample has weak evidence for a change in copy number, if other samples have the same deletion or duplication event, that can allow for accurate estimation of the copy number. So once we've estimated the aggregate copy number for each sample, now the next step is to basically figure out how many copies of each each paralog or the copy we have for each sample. And for this we have to rely on PSVs. So as PSVs are the only positions that differ between the different copies, And so if we look at the read depth for each sample at each PSV, that can tell us whether what's the ratio of the different number of copies given the aggregate copy number. So I'm showing you here 3 samples, A, B and C, and each of these samples has an aggregate copy number of 4, and I'm showing 4 PSVs, and within each cell shows the read counts, the allele specific read counts for each PSV and each sample. So what that means is that sample A at PSV1 has 15 reads for the yellow allele and 15 reads for the blue allele, and similarly for other PSVs. Now if I knew that PSV1 was a reliable PSV, which means that it never varies in the population, then because I see 15 reads for each allele at that PSV, then it would be safe to infer that sample A has 2 copies of the copy A and 2 copies of copy B, because the ratio is equal. But if I look at PSV2, I see 32 reads for the blue allele and 0 reads for the yellow allele. So that tells me that probably we have 4 copies of the copy B. So therefore, it's important to identify which PSVs are reliable and which are not, to figure out the parallel specific copy number. So this is basically, we have 2 sets of unknowns. One are the which PSVs are reliable and which are not, and then the other is the parallel specific copy number of each sample. So what we do is we model the frequency of the reference allele at each PSV, for each PSV and each copy. So that basically, this is a variable Fij, which is the population frequency of the reference allele for that PSV for copy I and PSVJ. And intuitively, if Fij is close to 1, that means that for all the different copies, that means that PSV is a reliable PSV, and vice versa. So this problem, the hidden variables are the parallel specific copy number, and we have these unknown allele frequencies. So it basically lends itself to to an EM algorithm to find the 2 sets of variables. So that's what we do. We basically use an EM algorithm to jointly estimate the parallel specific copy number and estimate F. And I'm just showing you the results from in this toy example. So sample A has parallel specific copy number of 2,2. Similarly, B also has 2,2, and sample C has copy number of 3,1. And we see that from the F values that all the PSVs are reliable except PSV2, because it has a frequency of 1/6 for for copy 1. So these are some results on the accuracy of the method for estimating aggregate copy number. In the interest of time, I'll go over them quickly, but we were able to find 10 genes for which there was experimental data on the aggregate copy number, and we found that our method had either better accuracy or equally good in accuracy compared to other methods. And we also used concordance analysis for samples with 2 different whole genome data sequencing data sets. And again, we found that for both aggregate copy number and parallel specific copy number, our method parascopy had very high accuracy. So I just wanted to show you something interesting about these the F values that I talked about, the PSV allele frequencies. So here I'm plotting the percentage of reliable PSVs across 83 disease associated genes. So, and there are 4 populations that we are showing here. So you can see that the genes on the left basically have no reliable PSVs, at least based on the EM algorithm that we used. And whereas genes on the right, almost all the PSVs are reliable. So we can see certain patterns here, that the PSV, the frequency of reliable PSVs seems to be more or less consistent across different populations. And also it's also consistent in most cases with previous data. For example, this PMS2 gene that is causes Lynch syndrome is well known to have extensive gene conversion, and there are no reliable PSVs in this gene, even though there are more than 30 different PSVs across the duplicated region. And kind of just going back to the SMN1 gene that we talked about earlier, that in this case we were able to find a small region where all populations except African populations have reliable PSVs that can be used to estimate the parallel specific copy number. And however, African populations show a different structure where there are no reliable PSVs. So for individuals with African ancestry, it's not possible to estimate the parallel specific copy number, at least using our method. How much time do you have left?
10 minutes more. 10 minutes more.
5 minutes? Okay. So I just wanted to close with a couple of examples of disease, like what can we learn about disease associations with these genes using our method. So NCF1 is a gene that's located on chromosome 7, and it's a gene that's triplicated in the human genome, and there are 2 pseudogene copies located to the left and right of this gene. This gene again is known to cause a severe recessive disease called chronic granulomatous disease, which is basically a severe immune disease that causes individuals have recurrent infections. And interestingly, the most common disease causing mutation in this gene is a PSV. So we have in exon 2 of this gene there is a dinucleotide sequence GT repeated twice, and the pseudogenes both have that deletion of GT, and therefore they cannot generate the functional because of the frame shift. And because of gene conversion, individuals the gene conversion between NCF1 B and C and between the functional gene can transfer the GT mutation to NCF1, and therefore individuals who have no functional copies of NCF1 get this disease. And using our PSVF values, we were able to estimate the frequency of individuals that carry only one copy of the functional NCF1 GT allele. And in this plot you can see here that almost about 3% of individuals in all populations seem to be carriers of one copy of this allele, and so therefore a significant proportion of individuals, about 2 in 10,000 are likely to have no functional NCF1 copy and probably have high risk of this disease. Another example is ABCC6. Mutations in this gene are known to cause again a recessive disease, and again this gene is triplicated, but it has a more complex duplication structure where only parts of the gene are duplicated. And most of the mutations that have been found are in the non-duplicated part of the gene, and using UK Biobank data that had 140 individuals with this PXE disease, we were able to find 6 individuals that carry deletions in the duplicated part of the gene, and compared to the individuals that don't have disease, this was a very significant association. So since I'm running out of time, I'll just just mention that our goal was to develop a set of tools that allow you to do variant calling, copy number analysis, as well as disease association in these genes, and we made kind of significant progress towards doing this, continuing to work on it. And our toolkit, one of the key design features is that it's highly scalable. It's a targeted method, so you can analyze thousands of existing whole genome data sets without doing any remapping or requiring large amounts of storage. And the pipeline for exome data is also in development, and the goal is to be able to enable novel disease gene associations, and also look at some existing associations more closely. So, and the hope is that others can also use these tools for studying genes of interest that have this duplication properties. So I just want to thank Timothy Prodanov, who led basically most of the work that I talked about today, and also Sang-Yoon and Yoon-jae, who are working on further developing this framework, and Bafna Lab and Gimrek Lab at UCSD, as well as the UK Biobank for allowing us to access the data. Thank you.